So hello everybody and welcome to the second virtual Grand Round at Great Ormond Street. I hope everyone can hear me okay. So today we are going to have a presentation from the infectious diseases team looking at the effects that COVID have had in children and really the story so far, because clearly it's an evolving story and something that is, we don't really know what's going to happen, how it's going to pan out. So it's going to be very interesting to see what their view is. They've got a number of people speaking to give us a range of different views and expertise, which is great. And just a brief thing about CPD, we have communicated with the RT-PCR who say that this sort of teaching event, you can self-register for one CPD point, which is 45 minutes of actual learning and 50 minutes of self-reflection. If you require certificates, you can fill out the feedback survey at the end, which you'll get a link to. SurveyMonkey and BGME will send you a certificate of attendance with your name on it. So that's pretty straightforward. So what I'll do is just briefly introduce Karen Moschel, who is our lead in infectious diseases, and she's been a consultant in infectious diseases at Great Ormond Street Hospital for the last 17 years, having trained in Cape Town, Imperial College London and Philadelphia. So she's been all around the world, and in 2004 she sets up an NGO in South Africa called Chiba, which is dealt with dealing with the HIV pandemic at the time in children, and that has since trained about 20,000 healthcare professionals. So she's well versed in the training and dealing with a pandemic such as this over the last 2 decades. So without any ado, I'm going to get you on to Karen, who's going to introduce our speakers. We've got Neela, then Charlotte, then Caroline speaking, and then we'll go on to the questions at the end.
Thank you very much, Jonathan, for that introduction. And hello to everybody. Thank you for having us and coming to listen to us. I'm going to speak very briefly before I hand over to Neela Alders, who is our Senior Fellow in Infectious Diseases, after which Charlotte Patterson, who's our Micro SBR, is going to be speaking. I really just want to take a moment to reflect on where we are and the wider picture before we narrow down and talk about our little piece and our contribution to the current pandemic. Pandemics are not new phenomena. They've been occurring for millennia, down the millennia, and they change the course of history, which might sound melodramatic, but is in fact the case, and they have a profound impact on our society. What is important for us as medical professionals is they also drive advances in medical practice and changes in the way we practice medicine very, very rapidly, which we are seeing today. Every pandemic is different, and this pandemic is very different to the HIV pandemic, which is the other one that has occurred in our lifetime and is still ongoing at the moment. However, a number of the advances, very rapid advances, particularly in virology, virological diagnostics and development of viral treatments, which were developed as a result of the HIV pandemic, are both informing and benefiting us in the current epidemic. We've changed the way we practice very quickly in our own institution. The way we manage patients has changed. We have an MDT management, which has harnessed social distancing and video conferencing to actually allow us to work much more closely and collaboratively together, which is an enormous advantage, particularly at the beginning when we are still unclear about precisely what we're dealing with. The PICU and the general pediatric teams have primary responsibility for patient care, infectious diseases, coordinating those patients, initial patient assessment, guideline and treatment development and implementation. Microbiology is contributing diagnostics, both the development and implementation of those diagnostics extremely rapidly, and infection prevention and control, amongst many other things, is ensuring both patient and staff safety and patient flow. We have contributions, significant contributions from a number of different specialties throughout the hospital, and I hope that you'll forgive me if I've left anybody out. It's inadvertent, but that includes immunology, hematology, cardiology, and rheumatology, and neurology is starting to see some patients, as I think are both the surgeons and the gastroenterologists. This is an introduction. I'm now going to introduce you to Neela, who's going to be talking about the clinical presentation of the first cohort of patients we have seen through our institution. It is the beginning of a story, and we will be continuing the story next week with the children who I know you've been hearing quite a lot about in the media. So thank you for being with us. Neela, over to you.
Hello, my name is Neela Aldis. I'm the Senior Clinical Fellow in Pediatric Infectious Diseases, and thank you very much for giving me the opportunity to present already our clinical perspective that we have on the GOSH in patients so far. So for the ones who were not able to attend the grand round last week, I just want to point out one thing so it's easier to understand the presentations. So there is the virus SARS CoV-2, which is responsible for the clinical syndrome that we are seeing at the moment. And we call that clinical syndrome the COVID-19. It is different from the SARS CoV-1 virus, which was responsible for the clinical outbreak between 2002 and 2004, what we called the SARS virus. We do know that all groups are susceptible, and what we already know that is in general that children are less affected. And so far we had 33 patients which have been tested positive on a respiratory sample, and as we say have a proven SARS CoV-2 infection. Of those 33 patients, 61% are male and 39% are female. So that is a bit similar to the numbers that we are seeing in other centers who have published their numbers. So of those patients, we can roughly divide them into three main subcategories, where one group is asymptomatic. So that means that we have picked those children up based on a screening, maybe because they needed to have surgery or they needed to have chemotherapy, or that they were unwell, but they had a clear proven other bacterial infection, which could explain the symptoms. And then there's a second group, which is the mild to moderate disease group, which reflects the children that are quite known to Great Ormond Street with significant comorbidities. We have been recently joined by the general Peds team of the North Central London cohort, and they have seen a couple of patients with SARS CoV-2 infections too. For example, there were two neonates who had a septic screen and were admitted in Great Ormond Street for further observation. And then there's a third group which contained 13 patients, which is slightly less than a mild to moderate group, which had 15 patients. And those patients are severely to critically unwell and end up on intensive care with different type of clinical pictures. So they often have acute respiratory distress, they need to be intubated and ventilated, they end up on inotropic support, but they do have other organ disease involvement, like for example, acute kidney injury, abdominal concerns, and they sometimes end up on hemofiltration. So it's quite a broad spectrum, and not all children are presenting with the same problems, but we see that a lot of different organs are damaged in the SARS-CoV-2 infections. And if we look at the background of those children, we see that some of them had some risk factors like prematurity, and one patient had a lower lobectomy. But in general, those patients are actually children who have been previously fit and well. So I want to wrap up with saying that I do think that we live in very interesting times. And to be honest, I feel quite privileged to experience this pandemic while working in a hospital like Great Ormond Street, where there's actually a massive amount of expertise available across different subspecialties, and where a lot of people are working who go above and beyond their ability to provide the best possible care to our patients in this rapidly changing environment. We so far had already 33 proven COVID patients where we have learned a lot, and it has given us already a lot of insight, but we do continue learning on a day-to-day basis, and where we saw initially the bit more typical pattern as one of them, which I've presented to you now. We do see a changing pattern, which children being admitted, we are quite hyperinflamed, but we will go a bit more into detail about that in our next sessions. So I really want to thank you for all your interest and listening to me. I'm now going to give the word to Charlotte Patterson from the microbiology team.
Thank you, Nila, and thanks for inviting me to speak at Grand Round today. My name's Dr. Charlotte Patterson. I'm a microbiology and infectious diseases registrar, and I'm going to tell you about COVID-19 or SARS CoV-2 testing, hopefully in around 10 minutes. I'm going to tell you about the types of tests we're currently doing at Great Ormond Street, so PCR and antibody, briefly mentioned viral culture as well, and how we're doing these tests at GOSH and how quickly we can test, and finally tell you about how many tests we've done so far. So firstly, PCR testing. This is known as molecular testing, and it's the mainstay of diagnosis for COVID-19. On the right-hand side of my screen, you can hopefully see the COVID-19 virus, a single-stranded RNA virus, which, of course, to perform PCR on, we have to extract and convert to DNA, and we undergo the PCR process, and at the end of that we get a CT value, this confusing value, a cycle threshold value, where high means low. So a typical strongly positive result would be around 15, and a weakly positive result around 40, and at 40 we have the cutoff where the assay becomes negative. And the target we look at is the N gene, which is different from the Public Health England assay. Ideally, this is taken within 5 days of symptom onset to give us the best possible yield and give us a positive result. And the type of sample that can be collected, I'm sure you're all aware, nasopharyngeal swabs, nasopharyngeal aspirates, by far the most commonly collected sample. And we can also test sputum, bronchoalveolar lavage samples, urine, stool, blood, cerebrospinal fluid, saliva. And so that's real-time PCR in a nutshell. The other type of testing, which is less commonly done at hospitals around the world, but we are doing one of the first hospitals to do it here on site at Great Ormond Street, is antibody testing, otherwise known as serology. And the presence of antibody suggests exposure to COVID-19. Really important to stress that doesn't necessarily correlate with immunity. So just because someone has positive antibodies to COVID-19, we don't yet know whether that means they are immune and can't catch the virus again. The antibody tests should be taken at least 21 days after exposure, although of course they may be positive sooner. And they look at the IgG and IgM response to COVID-19. Different targets are being looked at around the world, and at GOSH we're looking at the IgG to nucleocapsid, and rolling out a study looking at that in more detail at the moment. I briefly wanted to mention what is the significance of someone being PCR positive, and you'll notice this slide is about SARS CoV-1, so the original SARS virus that caused the outbreak in 2002 to 2004, and the reason I'm mentioning this is because we don't have that much data yet on what it means to be PCR positive for SARS CoV-2 and how long someone stays infected for. The graph on the right-hand side of the screen, the y-axis, you can see time, so that goes up to 21 days, and the x-axis, the percentage of specimens positive by real-time PCR. Fascinatingly, the thick black line at the top is stool PCR positivity, and you can see actually that over 50% of patients in this particular study looking at SARS CoV-1 were PCR positive still in their stool at 21 days. The dotted line underneath that is respiratory specimens, and around 20% of patients tested here were still PCR positive at 21 days, and urine at the bottom, none of their patients were positive at 21 days on urine PCR. So what does this mean? People are still positive, sometimes at 21 days, and actually for SARS CoV-2 up to 75 days after illness has been reported in stool samples. What does this mean? Well, we don't really know yet. This is a very small study published in Nature last month. Looking at just 9 patients, and these were adults. So again, we don't know too much about this in children yet, and immunocompetent adults as well. But this looked at active viral replication, so viral culture for COVID-19 in patients, and they looked at whether you could culture the virus at different stages in a patient's illness. For their 9 patients, not one single patient was culture positive after day 8 of illness. So although they remained PCR positive, they were culture negative, and the implications of that are that there's no active viral replication going on if someone is culture negative. Of course, this is all hypothetical, but this is what we understand from this. If there's no active viral replication, it's unlikely someone has actively replicating virus in their system still. And so they looked at the respiratory secretions and also the stool. And interestingly, in the stool, despite high CT values and samples that were strongly RNA positive on a real-time PCR, they never cultured virus from the stool. Of course, this was a very small group of patients, but fascinating results. And I'm sure we'll learn a lot more about this over the coming months. And so back to Great Ormond Street. So what tests are we doing at GOSH at the moment? Well, this is real-time PCR tests. We're doing 2 main pathways at the moment. The respiratory viral panel is the main one, and this it has huge scalability, so we can do hundreds of tests a day on the respiratory viral panel. Good turnaround times, generally within 24 hours and sometimes quicker. And we look at not just COVID-19, but at a wide panel of respiratory viruses. But the disadvantage is not as quick as our DiaCore, and the DiaCore gives us the quickest result, typically within a few hours. And gives us a wider respiratory panel of pathogens, including some bacteria like Mycoplasma pneumoniae. However, the disadvantages are that we have very limited capacity. Just 3 tests can be done at a time. And we had a huge issue with reagent availability. And originally, at one point, we only had 20 tests left in the hospital. And so we reserved this for very unwell new ICU admissions. And finally, how many tests have we done at GOSH? Now, this data is actually available to all of you via EPIC on the COVID-19 metrics and reports dashboard. And it gives us a really nice display of how many patients and staff we've tested and how many are positive. So on the left-hand side are patients, and the top line is individual patients, the lower line total number of tests done. I'm going to focus on the individual patients. So circled in red, you can see we've had 35 positive PCR patients, so just 4% of patients tested. And on the right-hand side of the screen, staff tested, and you can see we've had 200 positive staff, which is 30% of staff tested. And you can also see data on antibody testing on that COVID-19 metrics dashboard as well. So just to summarize, really, there's 2 main ways of testing for COVID-19 here, PCR and antibodies, and the mainstay of testing is nasopharyngeal secretions via NPA or NPS, but we can also test a wide range of other samples. There are different pathways at GOSH depending on what kind of turnaround time is required and the type of specimen. And with that, thank you for listening to me. I'm going to hand over to Caroline Dalton now, who's going to tell you about pharmaceutical treatments for COVID-19.
Hello, hi there. Thanks so much for having me. I'm just going to put my screen on full. So I'm going to really briefly talk about antivirals in the context of pediatric patients with COVID-19. Primarily focusing on our experience at Great Ormond Street, which has been with remdesivir, as Neela has mentioned. So this is quickly to say that we do have a local guideline in place. This was initially developed with our ID teams, micro teams, and a virology input initially. So the beginnings of this really started in January with a review of the existing literature at the time. And essentially it began as a bit of a dose-finding exercise, thinking that if this does kind of inevitably come our way, then at least we'll have a good idea of what to use and what kind of doses to use. But as the situation has evolved, as is this guideline, and at one point we were kind of updating it up to 3 times a day, and certainly it's still being updated really regularly, so at least kind of weekly. And it's since also been developed with input from our immunology and BMT teams. It's also been, it feeds in now to the British Pediatric Allergy, Immunology and Infection Group position statement, as well as the RT-PCR position statement, after developing this with colleagues nationally. So the most important thing really is that no antiviral has yet been approved in the UK for treatment of COVID-19. So despite having a guideline where, as Neil was saying, there's a lot of governance behind it, and we're certainly not advocating for blanket drug treatment for all paediatric patients with confirmed or suspected COVID-19. There's still not a huge amount of robust high-quality evidence available to support the use of antiviral medicines. So for now, treatment with any drug should ideally be in the context of clinical trials where they're eligible. And trials are being expanded to peds, and the team will be talking about those at Grand Round next week. So in the absence of appropriate clinical trials, it's sort of alongside appropriate, robust clinical governance when using a drug within the context of an available compassionate use scheme. And there I'm talking about remdesivir. So the remdesivir compassionate use scheme remains open to children who are less than 18 years old and pregnant women. And we've been able to successfully access this scheme to treat 5 patients at GOS. There's been a huge amount of governance input, as I've mentioned. So the ethics team at Great Ormond Street have been really instrumental in helping us access remdesivir for our patients. So please can I take this opportunity to say a huge thank you to Joe and Sarah and the rest of the ethics committee for their input and support throughout this process. And whilst this is still unlicensed in the UK, I'm sure you're all aware that after an emergency approval by the FDA, it's now licensed to treat COVID-19 in the US. So it's the novel antiviral agent under development by Gilead Sciences. And as I say, I can only speak to our experience at Great Ormond Street. We've had 5 patients treated to date, 2 neonates, 1 infant and 2 adolescents. So far, we've had all 5 patients who have improved, although it's difficult to tell whether or not they would have improved without the intervention of remdesivir at this stage. But what we have been doing is we've been reporting clinical and pharmacokinetic data back into Gilead to inform their teams regarding the use of this drug in pediatrics. And also to say that their team, and particularly one of their pharmacists, Neil, in the UK, has been really responsive. And as Neil was saying, that has meant that we've been able to get really rapid access to these drugs. So after local approval processes, of course. The treatment course is 10 days in total, so it's a loading dose followed by 9 days maintenance. And there's currently no advice to dose adjust for renal impairment, although it might exclude you from enrolment into the compassionate use scheme initially if you're in renal failure. And there's no adjustment, but it doesn't matter well, it doesn't matter obviously for the patient, but in terms of the use of the drug, we wouldn't dose adjust if that patient then began to deteriorate in terms of renal impairment. And there's also no dose adjustment for hepatic impairment currently. We've also had experience with dosing at extremes of body weight and have also been advised to use actual weight for these patients with an adjustment for neonates and the dilutions used, so an appropriate dilution to be used for infusion for smaller patients, including neonates. The main monitoring parameters that we keep an eye on would be, of course, the viral load and ALT, which is associated with potential toxicity and rapid rise in ALT, and of course, renal function, as you kind of would. So a little note at the bottom there is I'm primarily an antimicrobial stewardship pharmacist, so I've kind of got it in my head to not forget stewardship principles, and the rest of the team certainly agree with this as well. Support kind of drives our care. So most national guidelines are in adult patients in terms of antimicrobials with COVID. They reflect a constantly changing and emerging evidence base. So most patients at Great Ormond Street presenting with COVID will also be started empirically on some kind of antimicrobial treatment initially. But kind of remembering that pediatrics, as the team were alluding to, they seem to present with an atypical presentation compared to adults. So it's really important to not exclude other potential causes of infection before a filling investigation, hence the external MDTs that we do and all the consultations that we do prior to treatment approval with an antiviral specific to COVID. So kind of with our stewardship hat on, short courses where you're able to, and reviewing with microbiology and ID to ensure timely escalation and de-escalation of those antimicrobials. And then in terms of the antivirals, as I was saying, viral loads, we've been able to do them daily here, and contributing where possible to the pharmacokinetic data collection, as I mentioned, and feeding that back to Gilead. So I just want to flash this slide up at the end here, and a huge thanks to my colleague Joe Standing for putting this together for a previous webinar we've done. So he's also been absolutely instrumental in the development of our internal guideline. And he's put together this summary of the different drugs that I'm sure you've all heard bits and bobs about, particularly hydroxychloroquine over in the US, and now remdesivir over in the US, and also Kaletra. So here's the kind of overall main mode of actions of these medicines and a bit about the evidence. So as I said, it's changing really rapidly. So of note on this particular summary slide, the drugs in bold are currently featured in our guidelines. That's hydroxychloroquine, lopinavir, ritonavir, which is the antiretroviral Kaletra, and remdesivir. And there's interesting data coming out around favipiravir and potentially atazanavir as another antiretroviral used to treat HIV, potentially as an alternative protease inhibitor to Kaletra. So that's pretty much it from me for the time being, and I'd like to pass back now to Karen to finish off.
Hi everybody, sorry about that, and thank you to everybody who spoke. As I started this presentation with mentioning the different specialty groups that are involved in the care of all of these patients, I would like at this point, now that you have heard about the great deal of work that goes into caring for all of these children, to talk about the different staffing groups who have been responsible for the success of that care. The nursing staff, the allied health professionals, the middle grade and junior medical staff, and all the lab staff have had to change the way they do their work, and the volume of work that they've had to do very, very rapidly, almost from one day to the next. And they have provided the most extraordinary service. I know a lot of you are online at the moment, so would like to particularly thank you for that. At this point, we will now go over to questions and answers, but before that, just a quick advert for what we'll be talking about next week. The story will continue. It's a rapidly evolving story, and I know that a lot of you have heard about the hyperinflammatory syndrome in children, which we're calling PIMS-TS currently. We will be talking about that next week, when we hope that the clinical picture would have become clearer. A number of you working in the hospital will have seen these children and cared for them. So we hope that you will join us next week to hear more about that. And we will also be talking about research studies. So thank you all. I'll now hand over to Jonathan, and we will be joining others of our panel who are our infectious disease and microbiology team who will join in answering your questions.
Fantastic. Well, thank you very much, Karen, Neela, Charlotte and Caroline, for that really quite fascinating presentations. We've got a number of questions coming up. I'm just conscious of time. We've probably got about 10 minutes worth of questions here. So I'll try and group them into themes, as we did last week, to see if the panel can answer these questions. So the first one that seems to be relatively popular, really, is there a correlation between the BCG vaccination and COVID-19 and tuberculosis overall, which might explain some of the reasons why we're seeing such bad infections in some of the communities that might have latent TB?
That's a really interesting question. At the beginning of this process, about 2 months ago, there was talk of BCG actually being protective, and there's studies that were started in Australia to look at that and elsewhere. Certainly, if you look at the epidemic, or rather the pandemic in countries in Africa, places where BCG is quite prevalent, as well as India, they don't seem to be having the high death rates that we are having. Certainly, South Africa is 2 per million. It might have something more to do with the lockdown than to susceptibilities. But it's an interesting question that I don't think we quite have the answer to. Perhaps Alistair, Louis, and Delane, who are more on the research side, and Louis and Delane in particular, who have a TB expertise, would like to comment on that.
Thanks, Karen. I was just going to say that in many of those countries, there's a bias, of course, towards underreporting of cases as well. In those countries that have a high usage of BCG, so that's one thing. And then the other thing is to say there are large studies that are ongoing. I know colleagues at Harvard undertaking one in which they're looking at this kind of immune priming concept of BCG, in some ways protecting against COVID infection. But until those studies report and things, I don't think we can really conclude anything, and it's just a hypothesis at the moment.
Okay, anybody else got anything to say about that, or should we move on? That's pretty good. Okay, and there's a lot of questions about antibody testing. I mean, testing is what everyone's going on about. And so there's some concern about how sensitive our PCR tests are. And there's been some in the news that the North Middlesex test, for example, is only 60% sensitive. So there's a lot of false negatives. Do we know what our sensitivity is for the test that we are doing for our patients and staff here with the PCR test at Great Ormond Street? And does it differ hugely from the tests that are used widely by trusts around us?
Garth, do you or Charlotte want to pick that up?
Oh, so very sorry. Yeah, no, it's a very important question. We need to put this into context of how the test has developed over the last couple of months. The current test that we do here at Great Ormond Street it was developed really by Dr. Catherine Harris and Julianne Brown, is a very high performance PCR assay that tests the nucleocapsid gene. It has a very high performance, low cross-reactivity with other coronaviruses, and can pick up very low copy numbers of the virus in the sample itself. When the very first assay that was put out some months ago, which was the RT-PCR assay, suffered from some performance issues which may have reduced the sensitivity in the early stages. And I think these have largely been, we were very early and quick adopters of this highly sensitive assay. And I think that the reported and then you have to think about other issues that can affect sensitivity of test in patients. That's the sample quality, and also when you test the patients, because we know that the RNA can disappear from the upper airway in quite quickly after symptoms start, and therefore that may give you a false, in other words, you get a negative test where otherwise they might have been positive. And I think over the next couple of months, we will be, to get a true reflection of the sensitivity of the test, will require analysis of the serology and the clinical case definitions of the infection. It is not, I think, looking at the latest figures, we're looking at sensitivity of the current COVID N-gene assay, really, and it's really quite high, definitely in the mid-90s, with very little cross-reaction with any other virus. The only potential cross-reactive virus you could get would be the original SARS-1, which is virtually unknown in the world at the moment, so it'd be very unlikely that you'll get cross-reaction with that. So I think I think it would be unlikely now that it may be in the initial phases, the quoted sensitivities may have been reduced by some assay and sampling issues. I think we're very unlikely to see performance issues now with the current assays, including the commercial ones.
Great, thank you. Just one more about the testing. So I think what it looks like, you said that 30% of the staff you tested were positive. Now, how does that put us in comparison to other NHS trusts around us, or NHS trusts in the UK that are looking after children specifically? We high, low, we average?
I'm not party to some of the other figures. We're just looking briefly at the other partners. I would say we're just slightly lower than average for certainly for adult hospitals, would be quite a bit higher.
Okay, great. I think that's done with testing. So what we're going to do is move on to a little bit about the thromboembolic phenomenon that we're seeing with this. I mean, it sounds like with the hyperinflammatory, these children are at risk of thrombosis. Should we be looking at do anti-thrombotic deterrents in all children that are COVID positive, not the ones just demonstrating it, or the ones on critical care?
Thanks for that. That's out of our expertise, and we don't have one of the transfusion or hematologists on this, but I know that they are very involved with the kids on PICU, and most of them are on Delta-Par. There's also a recommendation that they receive vitamin K because of the clotting abnormalities when they come in with the syndrome. But we'll talk more about that when we talk about the hyperinflammatory syndrome next week. I don't know if Alistair or Louis would like to comment on that.
Yeah, just to add that we've been working really closely with Mary Mathias and her team, and they've set very early, even before we knew it, they'd set up a very well-running service where they would track patients being admitted with COVID or suspected COVID, would proactively have a message in Epic to alert that to the team that they need discussion with the anticoagulant team, and to guide very closely and daily, sometimes twice daily, the anticoagulant approach for these children. So that's been working, and we're learning more and more about that as we go along, and obviously learning from adult colleagues as well that have been dealing with much higher volumes of patients.
Thank you. I think that's done with thrombotic stuff. Talking a little bit about remdesivir, I think that people, that's probably not how you said it, is it? That people are very interested about that, and exactly a couple of questions that are coming up is, what is its side effect profile, and does it have any, if it's an analog of adenosine, does it have any similar properties to that?
Caroline, do you want to pick that up, or Alistair?
Thanks. I think all of us could probably pick that up, to be honest, because I think we're all in the same boat learning about this drug together. Yes, I'm a pharmacist, but I've certainly never, I mean, it's only just been released really, and it's been used previously in Ebola. So generally, what I can say is all of our patients who have had remdesivir have tolerated really well. I think there was one patient who had a slight increase in ALT, but I think it's really difficult to differentiate whether or not that was caused by the remdesivir or whether, there's kind of other things going on there as part of COVID overall. It's been really well tolerated, and we're, like I say, we're monitoring it really, really closely and feeding back to Gilead any adverse drug reactions. Alistair?
Yeah, that's exactly right, and I think to highlight that this drug has had compassionate access use within adult practice, and there's a paper with limited ability to be able to make any conclusions about efficacy, but it has data on tolerability and safety in adults, and it seems to be very well tolerated in adults. And the early trial data that's been partially released or released so far, randomized trial data, has also supported its safety, at least in adults. And so much so that, as Caroline mentioned, it's now been granted urgent license for adults with severe COVID in the USA. And I imagine EMA will follow suit quite soon. But definitely, even from the Ebola trials, there's no safety signal that has come out so far. On a slightly different note, it's quite similar to some of the other drugs, antivirals, that we do have experience of using, quite closely related to antiretrovirals, for example, some of the nucleotide reverse transcriptase inhibitors, and we are very sort of aware of their potential side effects and toxicity, and we'll be monitoring for that as well.
So, I mean, the obvious question, I suppose, would be, if it's got a very low side effect profile, is it why did we not use it earlier on in the disease process? Why are we waiting for it to become a very serious case of COVID before we use it? And then also, I think some people don't really understand what the compassionate use scheme is. We've never really heard of that before, and a little bit more of an explanation of that would be really helpful. Thank you.
That's a really good question, because that's a question that we've been asking too. Part of it is the the parameters that were set down by Gilead, which were driven to a degree by the studies that were set up. There are no studies in children, which is why we've been using them for compassionate use. And the legislation from those was that it would be used at a later stage when they enter PICU, rather than earlier on in the disease when it might have had greater effect. But it is also an IV preparation, and I think that again impacts on when in the disease process you use it. But I'm going to let Caroline and Alistair pick up as well.
I think it was also a little bit about making sure that we reserved it for the sickest of patients as well. So I think ideally with any antiretroviral, you want to start treatment really early, as Karen said. And actually I kind of alluded to that in one of my slides. So yeah, we should ideally be starting it much sooner, but it's an IV preparation. And also we were, I think, initially worried, Gilead were worried about supply and ensuring that we had supply for the sickest of patients. And also this is a situation that's developing constantly. So as we get more data and find out more about how which drugs work when, I think that's going to guide future treatment of this. In terms of what a compassionate use scheme is, essentially it's a way of accessing a drug for free by the company, and we kind of enter into a bit of a contract where we agree that we will use the drug in our patients because we hope it's going to help them, and then we'll also feed back to the company and help them develop, kind of increase their data and help them kind of develop the drug for marketing in certain indications in certain patient cohorts. So we access it quite a lot with medicines that aren't necessarily licensed in this country or aren't necessarily licensed in the indication that we're using it in. And that's an exchange of kind of getting access to the drug without any additional kind of economic cost. We feed back to the company with our experience of its use.
You just mentioned economic cost. I mean, what is the cost of a course of this drug?
It's not marketed yet, so I don't know.
Do you know what it's likely to be?
No, I imagine if they show it's useful, it'll be very expensive. But
thinking about buying shares, that's
comment
whether it's worthwhile.
Yeah, I couldn't comment. But it would be really interesting to see. Kind of when we do, we'll have a bit more data with how it's going to be used in the future and what it's going to be useful for. It's exciting.
Can I just add that we're heavily involved with advising, giving clinical advice to Gilead on setting up phase 2-3 clinical trials in children as well, and we hope to be a site and actively involved in that.
And actually, we've informed some of their protocols for the smaller patients. So I think our first patient, they're typically a premature neonate, which is obviously a pharmacokinetical nightmare to a certain extent. Very little evidence for these drugs in any pediatric patients, never mind a premature neonate. So actually our data then ended up feeding into the Gilead protocol, and that ended up, we ended up getting different advice and we changed their protocol, like I said, for the dilutions. So yeah, it's been a really, really good working relationship, I think, with Gilead, and a good back and forth and yeah.
Okay, great. And I'm just conscious we are running out of time, sadly. But so one more question. I think there's been some concern from people that have got patients that are immunocompromised, immunosuppressed on chemotherapy. Is there? I mean, the advice for this is to shield them. I mean, do we need to look after them in any more ways than we're actually doing at the moment? So are we seeing an increase in infection in that group, or an increase in severity of the disease once they're infected with it?
No, and that's what's really, really interesting. They certainly don't need to be shielded beyond what has been advised. And we've got frequently asked questions for all of those groups on the GOSH website, which have been developed in conjunction with us as a team, giving that advice. No, we have not seen an increase or more severe cases in the immunocompromised group, whether it's hematology, oncology, BMT, gastro, rheumatology, immunology. We have seen the occasional patient from cardiology. However, it's probably a reflection of the general population rather than that particular group. There's no evidence at the moment to suggest that those children are any more in danger of severe disease than any other child currently. Don't know if anybody else.
One more comment. Would immunosuppression actually help with the course of the disease if it's a pro-inflammatory state, which is causing the symptoms?
That's the obvious question. I think that we're going to take the fifth on that because we just don't know. We are using some of those drugs to treat it, but I think it would be very dangerous at this point to say that they are protective. I think that we need to look after this group the way we've always looked after them. They're a vulnerable group and they need to be isolated or shielded depending on what their specialty teams feel is most appropriate. And I don't think we should be either falsely pessimistic or falsely cavalier about how they are looked after. Jonathan, I know that you're probably going to close, but can I ask you to give Louis 2 minutes just to have the last word?
Yeah, thanks. I just wanted to take the opportunity to highlight the CoSTAR study, which has just been set up. The Health Research Authority gave us rapid approval to do this study within 12 hours. I don't know if that's ever been approved that fast before, so I'm really pleased with that. And the clinical research facility staff have been incredible at helping this get up to speed and start running. Within the first 4 days of recruiting, we've got coming up to 400 participants already. We've only opened so far to the caterers, the porters, the cleaning staff, and the ICUs, with a few other individuals filling up the slots to keep us busy. And we hope to be opening to the whole trust fairly soon. We're mindful this needs to be open to everybody as soon as we can feasibly get the study open. The study is to look at our all GOSH staff antibody levels, and we've got a decent test that we're using that we've tested in-house and checked the sensitivity and specificity to do this, because we think it's really important that we all know our status, even though we're not certain of whether that confers immunity or not yet. And we also need to know, and this is really critical, how quickly those antibodies wane. Okay, so if any of you are out there, and we can't approach you individually, we have to do it as part of a generic trust email. If any of you are out there who know that you're antibody positive, please get in touch with us or come up to testing, and we will get you involved, because it's really useful to try and know what is happening to your antibodies over time, and whether they just disappear, and whether this notion of herd immunity doesn't exist. So it's just a shout out to those people, and also thank you to all of the CoSTAR study staff who have been really fab so far in getting it off the ground.
Okay, thank you very much, Louis. Well, look, I just want to say thank you very much to Karen and your team, Nella, Charlotte and Caroline, for their presentations, and thanks for the panel for answering the questions. So well. You're all going to be back, or some of you will be back next week, which is Tuesday, which is our next grand round, same time, 1 o'clock, and the same process through a webinar. The details are there. The adverts will come out. They're going to be on email and on screensavers. Slido questions, we reset Slido, so we'll get more questions for next week to be relevant to the talks. And we're going to say about, if you would like to get a CPD point from the RT-PCR, you can self-certify, as we said at the beginning. If you need to have a attendance certificate, so you can email, complete the survey for the feedback for the session, and then we will email you an attendance certificate with your name on it to use for your appraisal if needed. So fantastic. Thank you very much, everyone that took part. We had 457 participants at the peak, as I saw it on my screen, which is fantastic. I mean, that is the biggest grand round we've ever had, which would fill the Kennedy Lecture Theatre twice over, actually, if you can imagine that, filled twice. And so that's the biggest group of people I've ever spoken to in my life, I can tell you. And you guys did a fantastic job doing that. So thank you very much for your input. And we'll see you next week, on Tuesday next week, okay? Thank you.
Thanks.